For stress test, at first I put about 5 ml ester solution in a boiling tube. Then I add 1 drop iodine solution. 1 drop iodine solution is enough for stress test. We see that solution turns blue-black. For reducing sugar test, I put about 5 ml reducing sugar in a boiling tube. Then I add equal volume of Benedict solution. Now I will keep this solution in a hot water bath about 5 minutes. Temperature should be more than 80 degrees centigrade. For protein test, I put 1 spatula albumin powder in a test tube. Then I add about 2 ml distilled water and shake well. Now I add 4 to 5 drops Biret reagent. We see that solution. We see that compound turns purple color. For fat test, I put about 1 ml oil in a test tube. Then I add 2 ml ethanol. And shake vigorously. Now I will keep this for 3 minutes. Before this, I add 2 ml water. After 5 minutes, we see that reducing sugar turns blue to red brown. Color depends on concentration. Low concentration turns blue to yellow. High concentration turns blue to orange or red brown. After 3 minutes, we see that fat produces emulsion. Oil is in upper portion and the rest of the compounds in the lower portion.